Hey everybody, thanks for having me today. It's really great to be a part of this symposium. It's just a fantastic group of people together, and I heard yesterday went great. I was in clinic all day, so I didn't have a chance to get out, but it's just really happy to be here. So I've been doing some work with a huge team on the urinary microbiome. We're really in our preliminary studies with this. We've had great support from Karen, Paul, James in the immunology lab, and Dirk Lang is a collaborator who does a lot of microbiome work with kidney stone disease in our urologic department. We have a collaboration with Cleveland Clinic, a guy named Aaron Miller down there who does a lot of microbiome work, and a lot of the samples we've sent out has been analyzed down there as of now, but we're hoping to actually use nanopore, which is going to be talked about later on today, to do some in-house analyzation ourselves. So we're really at the preliminary work in what we're doing here, but I just wanted to share some of that and hopefully get everybody a little bit excited about analyzing what's in the pee. Okay, the topic is urinary microbiome an innocent bystander or active participant in kidney allograft dysfunction? I'm a transplanter, and everything I think about is transplant related. And I'll spoil it right now. I don't have the answer to this question. I don't know, but hopefully with years and years of work, we might come close. All right, so Human Microbiome Project was established by the National Health Institute in the US in 2007, so it's been going on for a really long time. The whole goal is to comprehensively characterize the human microbiome, which includes bacteria mainly, but also viruses and everything else in between. What they found is there's no question, all throughout our body, we have diverse microbial communities. And there's these commensal organisms that live in symbiosis with us, but in pathologic states, there's alterations. And this has been proven for multiple disease states. So I'm just going to do a little bit of a background because a lot of people aren't familiar with this particular line of thinking. So there's three pictures that have been brought up here, and these three pictures represent the same population. All we're doing is we're using different laboratory techniques to analyze these three pictures. Microbiota, that's 16S RNA sequencing that we're doing. It's a great technique. And that just tells you what bacteria are present. And that's about it. When we look at the next picture, that's saying what genetic material is present and what is that genetic material capable of doing. That's looking at the metagenome of these bacteria. And then the microbiome, that constitutes the metagenome, but also all the products of these bacteria and all the pieces of these bacteria. And that's a whole different and more elaborate and more expensive way of looking at this data and characterizing it. So what's clear, what scientists have definitely found is there's numerous bidirectional interactions with the host to alter organ function, tissue permeability, and immune system responsivity. These microbes in our body, they do stuff. And it's either, it's a chicken and egg thing. Is it them that's influencing the body, or is the body influencing them to change? We don't know these answers. These are some of the laboratory techniques just thrown up to see where we started. We started with traditional culturing, and then we went into this enhanced culturing, which is actually taking place in most microbiologic labs nowadays. And then there's, of course, the genomic sequencing, and each of these increases your sample resolution, but also the phylogenetic resolution. And phylogenetic resolution, what that implies is what's the whole environmental picture? What's the big picture that's going on here? And that's really what we want. We want high resolution and high phytogenic resolution. And we're not quite there yet. So we now know the urine has bacteria present in it. 5-10 years ago, we thought it was totally sterile. So this is a new way to think. Clearly, the GI, the gastrointestinal tract, has been extensively studied. And the urobiome has been, there's very infrequent studies, especially in transplant patients. There's a handful of studies, and I'll present some of those for you guys. So GI, let's look at GI first, just to get a sense of what they found there, because maybe we might find some similar things in the urinary tract. The GI, the bacteria have been found to have protective functions, metabolic functions, and structural functions. There's a little diagram there sort of highlighting some of the immunologic functions that these bacteria have been postulated to produce. But clearly, these functions that they serve in the GI tract, probably there might be some sort of similar characteristics in the urinary tract, even though the urinary tract in general has different functions than the GI tract. GI tract is much more dynamic than the urinary tract, where the urinary tract is much more of a reservoir. But everything in the urinary tract is produced by the kidney. And the kidney is what I care about, as you can see by the tie. It's a kidney tie. And clearly from all this GI work, they've got some emerging therapies. And these therapies are real world therapies that have come about from this research that has been conducted. And it sort of blows my mind that nobody's focused on the P yet, because in great detail, because everything in the body is connected. This homeostasis of the body, I'm a big believer in it, and it's a good thing. So we've gone from the gut. Now there's a so-called gut-kidney axis. When the gut is not functioning optimally, that can produce damage to the kidney. And there's certain markers that have been shown in chronic kidney disease and other kidney-related issues. But some postulated mechanisms for how these things come about is there could be reduced tight junctions from this imbalance of pathogenic bacteria and the commensal bacteria that help us out. There can be this increased trimethylamine oxide that's produced by these bacteria, and it gets into our circulation, and it's very associated with chronic kidney disease. There's other uremic toxins that are produced where they can't be eliminated properly by the kidney and they build up. Short chain fatty acids are another big sort of marker of dysbiosis that they're looking at. And composition, I'll just focus on that bottom, the composition, this decrease in alpha diversity. Alpha diversity implies the diversity of species within a population. Beta diversity implies the composition of that population. So if you have one specimen that has E. coli only, and you have another specimen that has Pseudomonas, they have completely different beta diversity. They're completely different composition. And that's something that in our work, we're really looking heavily at. Women are an important person to think, because women have a beyond a gut kidney axis. They almost have a gut kidney vagina axis, because all of these things are intimately associated. And you can see the urinary microbiota and the gut and the vagina, they do share some species together, but they also are completely different in steady state in women. This was a really nice study that was looking at urinary tract infection. And I put up this slide not to think about urinary tract infection, but I think you could almost replace any single disease state where that urinary tract infection is, and these interactions could produce that disease state. So let's say chronic kidney disease is something, or transplant rejection, or chronic allograft dysfunction, increase IFTA, whatever you want. That can probably all go where urinary tract infection is, but that could all come about with alterations in the vaginal mucosa. It could come about with alterations in the urinary microbiota or the intestinal microbiota. And all of these things change with age. They change with decreasing hormonal levels, different environment, different diets. It's a very complex topic. And I hate to make it even more complex, but now we got transplant factors that are mixed into this whole thing too. So we got recipient-related factors, donor-related factors, and transplant management-related factors. So clearly, we can't look at the microbiome in isolation. If we're doing that, we're missing the whole picture. It's very much like that great talk before in the ICU. You know, it's there's multifactorial things that lead to these patients and the distress that they get, and we always have to have that in the back of our mind. Sunny Yang, who's been working really, really hard with me, has done a review on the microbiome, and we've just sort of highlighted a few of the different areas we're talking about, the GI, the vagina, and the urine in non-transplant patients, to see what they've found. In general, healthy state, they have a certain composition of microbiome that's pretty steady across fields in the GI tract. When antibiotics are introduced, there's decreased diversity, and the composition changes when regrowth. So change in alpha diversity, change in beta diversity. And that's pretty much a consistent finding throughout all of these studies. Okay, decreased diversity, change in composition. Okay, the urinary microbiome now, let's focus on that. We're finally there. Okay, so there's a rich species variation in the urine. We know this without a doubt. But it does change over time, over place, and it's sort of hard to know what to do with the information when you get it at this point in time. I think the urine is a great thing to study because, like I said, the kidney produces it and it's in contact with the kidney. And whether it's the urine influencing the kidney or the kidney and the systemic functions influencing what's in the urine, it's debatable at this stage because we don't know. I love it that it's easy to get, and there's potential for liquid biopsy with the microbiome work, but of course, there's many other things: cancer detection, kidney allograft dysfunction, a whole bunch of other different markers and proteins are being looked at. Is this a liquid biopsy of kidney health? And this is just one avenue of thought. So in transplant patients, it's really not investigated well. I always, when we're thinking about microbiology, we have to think about how these specimens are being collected as well. Unfortunately, it's really hard to collect things and then analyze things in a completely sterile fashion in real world clinical practice. So there are always going to be confounders. And I think there is a huge risk of contamination in this type of work. So there's some laboratory studies you can do to try and cancel that out. Just highlight some postulated roles of how bacteria may help your urinary tract and your kidney health. If you look at number one, those could be neurotransplanters influencing the immune system arising in the urinary tract. Number two, the bacteria could get those harmful, the commensals could attack the pathogenic bacteria. Number three is reinforce tight junctions, keep that barrier intact. Number four there is gobble up the peptides and the dangerous substances that are produced in the urinary tract. Number five, and I love this, that this points to the kidney, that's interaction with the mucosal toll-like receptors and cells, which can produce subsequent downstream inflammation. So in kidney transplant patients, when we set about this review, there was 5 studies we found totally looking at the urinary microbiome in kidney transplant patients. There's a couple more now, but there was 5. That's unbelievable to me when this thing has been around since 2007. And really and truly, all of these studies are 16S RNA sequencing. So we're just looking at the microbiota. We're just looking at what's there. We're not even downstream of that doing the metagenomic analysis, and we're not even doing the shotgun analysis where we're really understanding the multi-omic nature of what the effects are on these situations. So in general, I think the last 3 are the most highlighting. Again, decreased diversity of specimens, change in composition. And I really find it funny that when you look at the stable disease state, the characterization of the urine, it says increased diversity and decreased diversity. And the reason it says that is because they were analyzing different populations, and different populations in different environments produce different outcomes with this. So clearly, we don't know what a stable post-transplant state looks like with the urinary microbiome at this point. Does anybody have any questions at this point in time? That was a background, because I think a lot of people don't know much about this, and maybe I'm wrong. Maybe everyone's an expert in this, but I feel like it's a new topic in some ways. Let's keep going. All right. So the research questions that we had. So we'll get into 2 of the projects that we did, which have a lot of little projects. So we want to know what's in the urine. Can the alterations in the urine hint at disease states? Can urine bacteria play a role in shaping the inflammatory environment? This is what we want to know. So we have a renal transplant clinical specimen environment that Karen and Franz have been instrumental at getting me some specimens for. And then Sunny Yang, who did some great work, looked at bacterial components and what happens on cultured kidney cells with those. So we had 42 patients, 86 samples. It was a midstream clean catch urine. So already there could be contamination. It was biobanked. So it all had to get processed. So already there could be some contamination in the whole thing. But the good news is all specimens were handled in sort of the same way. So that sort of can unify things for you. So we looked at prior to transplant, at the time of an inflammatory event, and in the particular specimens we had, that was some form of rejection or borderline rejection. And we looked at 1 month post-transplant and 3 months post-transplant to see how things change over time or change with an inflammatory event. This was just a 16S RNA sequencing. We have some ongoing work doing some shotgun sequencing on these samples right now, which is should be coming out pretty soon. And we looked at alpha diversity. Again, there's a number of species in the specimen, and beta diversity, the microbiome composition. So our results, we had early rejectors. Most of these were men. We had women less than 50, women greater than 50, because microbiome changes with age in the studies. Men less than 50, men greater than 50, because the man's prostate grows with age, as many are aware in here, and that can change the microbiome with urinary obstruction. A rare fraction analysis is just saying, do we have a richness of species to even conduct these studies? And all the samples had enough richness of species. And following this, we underwent a decontamination step where we removed some of the ones that might be contributing factors, the confounding factors where they got contaminated during processing. Just going slow. Oh, I might have skipped a couple of slides there, but the taxonomic profile. So we had a negative control, which was based on some local Cleveland Clinic patient specimens. We had a positive control. That was urinary stone disease was our positive control, and our samples. And as you can see, our samples had distinct beta diversity. So there was a different composition from the negative and the positive control, but alpha diversity, alpha sampling was standard and non-significant. I think these are big files, so they just take a while to switch slides. The next, we looked at the taxonomic profile. Sorry, guys. Technical difficulties, but it's, we'll go through it pretty quick. I think it's just a file. There we go. We had some rejectors. So essentially in the rejection sample, we just looked what was there, and these rejectors had basically normal flora that we would see in anybody, really. They didn't have any real distinct bacteria, and it was pretty much what you would see in anybody. So pre-transplant comparisons, no rejection versus rejection. Again, in these samples, again, I want to focus, we're looking at the cohort here, the whole cohort altogether. So we're not looking at the individual level at this point in time. There was no difference on alpha analysis or beta analysis in no rejection versus the rejection specimens. Effects of transplant, so pre-transplant to 3 months post-transplant, how did things change? In our samples, there was no difference in alpha or beta diversity at this time either. Effects of rejection. Again, at a cohort level, there was no difference in the effects of rejection in our sample. Age and gender. So gender, we clearly saw a difference in the composition of the urine, as we would expect. Women's bladder composition is different than men's. And age-wise, we didn't see any difference. So older women or younger women in our cohort, or older men and younger women, there was no difference. But when we look at the individual level, now here's something pretty profound. What we found in both groups is that the beta, so the change in composition, pre-transplant to post-transplant at all time points, was drastically different and highly significant at the individual level. So greater than 75% of the bacterial composition post-transplant was changed in our samples that we sent for analysis. Now, that's profound. Aaron Miller, who works on this and does a whole bunch of these studies, has never really seen individual variation greater than 25 to 50% over time. So this is a very unique finding, and transplantation is a very unique thing where we're putting somebody else's tissue into another person. These patients get antibiotics, these patients get immunosuppression medications, in addition to other managements. And exactly why we're seeing such a profound change in composition at the individual level is unknown at this time, but we're going to find out one day. So at the population cohort level, there were no consistent differences. When we look at the individual level, again, it's a 75% change, very, very profound change. We wonder, could some donor microbiome be transmitted across? And we have some future studies planned for this. Is there another reason for this finding as speculated? And ultimately, we're doing some ongoing work right now to see if we can actually tease out exactly what we're seeing based on the samples that we have right now. So bacterial component study by Sunny Yang. She did a great job looking at LPS and LTA, which can be virulence factors of bacteria. We looked at these with cultured kidney cells, mesangial cells, and proximal tubular cells, and there's a diagram just to show where those are located. The results, I think, are fantastic and very interesting. When LTA and LPS were inoculated with this tissue, we looked at their metabolic rates of the specimens, and the tissue with LTA and LPS alone showed decreasing metabolic rates. But when you combined LTA and LPS together, the metabolic rates were actually preserved. That's a green line that's higher, and these were significant. So there was some sort of modulating effect when both of these were put together. We have some cytokine analysis ongoing, and in this heat graph, I mean, basically, if you look at the top, you can see LTA, LPS, and LTA, LPS categories, and you can see that the heat map is completely different across the boards. And what we're finding is there are some key cytokines and key genes that are upregulated in the single groups, but then downregulated when they're combined together. Exactly why that is, we're not totally sure, but we really want to try and uncover the mechanisms for why that might be. So conclusion of this, LPS and LTA both act as stressors. Combining them led to a reduced metabolic influence and a reduction in pro-inflammatory genes. So there's something there. And ultimately, I want to convince you that this is important work, and more work needs to happen on it. So I'll just leave you with 2 minutes here of a paradigm shift. You know, when we see bacteria, we think it's bad. We treat it with antibiotics as clinicians right now. And there is a little bit of a switching of thinking that maybe we can modulate this so that we don't get these multi-drug resistant specimens. And maybe there's some different therapies we can employ rather than antibiotics itself. We've always been focused, bacteria bad. Well, actually, it's not true. We got lots of good bacteria in us, and we want to harbor those, and we want to nurture those good bacteria. Potential therapeutics, again, on the left-hand side, that's where we are right now. On the right-hand side, that's where we want to go. We want to get some host microbiome modulation. There's lots of different postulated ways that we might do this. None of these are prime time right now. None of them have good evidence behind them, but all of them warrant further investigation to see if something can actually help patients. Just to highlight some of them, if you look down the host microbiome modularization profile, probiotics are probably one of the most extensively investigated things to date, and studies don't really show much benefit to those time and time again. They're talking about doing attenuated E. coli strains, either catheter coating those, inoculating those into the bladder to see if those can outcompete the pathogenic bacteria of some folks. And there's no good evidence that that works. And to try and actually run that as a clinical trial is going to be very challenging when it's all said and done. So there are a lot of challenges there. And then again, we talked about the GI, vagina, kidney access. Could we inoculate other therapies into some of these locations and see an effect in the urinary tract? We don't know yet. So a lot of these things are yet to be determined, and there's a lot of other thoughts as well. But I'm happy to take any questions. But I think I've convinced we're just at the tip of the iceberg. You know, no symposium is complete without somebody throwing up an iceberg slide. And I'd be happy to take any questions. And again, transplant's the ultimate team game. We're all we all work together to figure it out, and I really like that slide about how we're figuring out the clinical side and the basic science side, because it is a really challenging thing, and it just takes collaboration and getting coffees together and figuring out how you can make things work, and because these are all resource intensive projects, they cost a lot of money, so you got to work together to make it all happen. Thanks, guys. And I may not have all the answers to the questions right now, as you can see.
I have a question here. So I think you should in cohort level, there's no difference, but in individual level, there's a big difference like before and after transplantation. So is there any, maybe I already missed that, but did you have any results regarding to what makes the difference between the rejector and non-rejector? Or like, what's the difference in the before the transplantation? Is there any clue that you can find to predict that it can be based on the microbiota, this one can be have rejection and the other one not? Yeah, we did pick out some specific phylums and genus of bacteria that were a little more heavily included in the rejector group compared to the non-rejector group. The rejector group was mainly men. We had 9 men, 1 woman.
And one of the bacteria that came out on a sub-analysis, which I didn't include here because we don't really know how to interpret it yet. We're trying to put our heads together to figure out, but it was Corynebacterium. And Corynebacterium is a typical skin bacteria, and it seemed to be a little more prevalent in the rejectors compared to the non-rejectors. It's not really thought of as a pathogenic bacteria in any stretch of the means, but for whatever reason, it was more highly represented in the rejector groups. And that's also one of those bugs that could be more a skin bug that's more of a contaminant than anything else too. But it's interesting that it was more prevalent in that group. So yeah, we have a lot of like sub-analysis where we're trying to really figure out what all these things mean. We just got this data a couple of weeks ago, actually, so it's hot off the press. But there's a lot of sub-analysis we're doing. One of the problems, and Karen can speak to this as well, is our specimens. We don't always have perfect specimens for everybody either. Not everybody gives a specimen at certain time points. And I think that's why we sort of have low numbers of rejector specimens when it's all said and done. And I think if we did this on a broader scale, we might see very different results. So it's just really a pilot study at this point in time, and we need more information. But yeah, there is some stuff that we're gonna look at and see what's the deal is.
Great talk over here. And you already mentioned probiotics as potential therapy, but is there sort of a movement starting to happen towards just generally dietary interventions pre and post transplants? Because we all obviously know that certain diets cause low-level inflammation, and then others can really have anti-inflammatory effects, and probably that can greatly impact, depending on what your position was before the transplant and after, how you might fare like keeping the transplant.
Yeah, I mean, that's a fantastic question and point. And to the best of my knowledge, there's nothing like that going on in the transplant community. And that is something that we're really, really interested in doing. Dirk Lang, who I work with a lot on this, he does this in the kidney stone population. He's already doing dietary interventions in that group. And our hope is over time, we're going to roll that out to our transplant patients as well. So yeah, it's there's about like 100 studies that are sort of like being thought of, and we're just figuring out how to implement them. Really, that's the hardest part about this whole thing, like you've alluded to. But yeah, that's a great point.
Thank you.
Hi, Dave. I have like 600 questions. I've already forgotten the first 100 of them. But one question was, actually, Rob and I were just chatting, whether or not the immunosuppressive management these patients were on would affect, I mean, I'm assuming that they must do, but how you include that in the study?
Yeah, I mean, we're definitely going to look at that, and we're going to see, we're going to put them into different groups. We're like, once we get all the clinical variables put in, we're going to put them all into subgroups, and we're going to analyze what we're seeing in the bacteria. Like, almost all of our patients are going to be induced with IL-2 induction, I suspect. I think there'll be very few thymoglobulin inductions in the group. But that's a question that I would really have. You know, is T cell depletion versus just blockade, is that going to show a difference in what we're seeing here? And I think it will. I think if we have enough samples, that it will. So it might be that Giuseppe, who you saw give a talk yesterday, we might collaborate with Wake Forest to try and increase our samples and to try and see if there's a difference there, because that's actually what I'm most interested in. I'm most interested in how the current clinical management affects things.
Rob's like, ask him about withdrawing immunosuppression. So whether or not if you withdraw immunosuppression, is that going to change the way that the microbiota basically respond? Does the microbiota respond to immunosuppression that's being withdrawn?
Yeah,
probably.
Yeah. We don't know. I mean, we might see with some of our patients, we drop prednisone over time, and we do a prednisone tapering. So maybe we'll see an association with microbiome change over time, or we're going to look at it. Yeah.
And sorry, two small other questions. I have so many questions. For the women, I mean, you had less women in your cohort, but maybe when you think about designing it down the line, making a note of when these women are menstruating, because I've seen studies that show that the microbiota of the vagina, and you have the axis of the vagina, the gut, and the urinary tract, that actually changes quite significantly at the time of ovulation and menstruation, actually.
Yeah.
So, you have the age control. So I think actually that fits nicely, because some of these women won't still be menstruating, but then the younger population, you may have to collect that information as well.
Yeah, absolutely. For prospective studies, we'll definitely have to collect that. That's a great, yeah. I think I have one more question and then we'll head off. But I'm happy to talk after because I love these questions because it means you guys like it.
That's still on in the middle.
Yeah. Are you hypothesizing the microbiota or what they can make it and excrete in the system? So in case of E. coli 0157, how they can dissolve the kidney with hemolytic uremic syndrome.
Yep.
Something can be Shiga toxin or something can be the component, biomolecular component that is affecting kidney indirectly. So in that case, you are dealing with a huge amount of information of individual microbiota. So how are you planning to do?
I think the analysis is that we want to understand the whole microbiome. So we want to understand the bacteria that are there and all the components that they produce. And the data I presented now is just the 16S data, the metagenomic data, and the microbiome full shotgun data. That's actually going to be ready in a couple of weeks from now. So we don't have that data yet. So I don't know how to interpret it or really how to answer it, but 100%, you have to look at the whole picture. You have to look at the environment and everything else. And I don't even know if that'll tell the whole story because there's so many other clinical and other variables that go into it too. So to try and put this small little thing in the big picture is a really hard thing to do.
Yeah. Okay.
Yeah, but it's a great question. Yeah, no, for sure. All right. Thanks, guys. Really appreciate it.